bonjour, plutôt bonsoir à tous. Donc on continue dans ce cycle de webinaires de la FHU PASSEM. Donc je suis Karine Clément et je vais co-chairer avec ma collègue que vous voyez à l'écran, Nathalie Fontel. Donc merci Nina pour l'organisation. Et donc pour démarrer, donc aujourd'hui on va entendre Lejla IMAMOVIC qui donc a un background en microbiologie, qui a fait un postdoc en biologie des systèmes, aussi microbiologie, et qui a rejoint le laboratoire de Guy Rochoc en tant que chargé de recherche à l'Inserm, donc au CIMI autour des maladies, en particulier déficit immunitaire. Et donc elle va présenter son travail en anglais sur le virome dans les pathologies, et donc ce domaine est en train de se développer à grande vitesse en fait, et donc voilà, Lejla, maybe you can start, et donc on rappellera donc après la présentation les futurs événements. So I guess Lejla, the floor is yours.
Thank you very much. So thank you for this opportunity to present the work on virome, on that virome we are doing at the Guy Rochoc team. So gut microbiota represents a complex ecosystem that is composed of both prokaryotic and eukaryotic microbes, but also viruses. Bacteria, of course, are the most dominant, but some estimates also put viruses ahead of bacteria, which is still currently under review. So gut viruses are composed of eukaryotic and prokaryotic viruses, and prokaryotic viruses are largely dominant. More than 90% sequences are belonging to these viruses. Collectively, they are called phageum. Eukaryotic viruses are represented by viruses that infect microbes, eukaryotic microbes, and humans, and also viruses that infect plants, which probably originated from food. So viruses are also highly diverse biological entities. This is somewhat visible by the fact that they don't have common origin. They can be both double-stranded and single-stranded, and both with RNA or DNA genomes. In the gut, we know that class Caudaviracetes is somewhat dominant, but gut virome is still largely unexplored part of microbiome. So this is obvious by the discovery of novel phages on a very frequent basis. The most drastic example of this discovery was 9-10 years ago, where a novel phage called CRAS, based on the cross-assembling of human reads from gut-imaged genomes, was discovered. So this was novel phage, highly abundant in 75% of global human gut, and was largely also abundant. Now, this novel phage is nothing new. After this, several novel phages have been discovered. And maybe the landmark study of this novelty of phages was a paper that collected all metagenomics data and identified novel viruses, over 140,000 new species in human gut, and half of them were assumed to be novel. So when it comes to gut virome, this novelty is also referred to as viral dark matter. And not that we only don't know viral species, often even if we can identify the virus, we have difficulty saying what this virus is encoding. So just to illustrate this, I'm showing you the virus, for example, that we saw in our recent study. So this represents 100 kb assembly, and only a small fraction of this assembly we can actually identify as some phage-like proteins. The rest we don't map the database. So this inability to map the database, to discover the function and what these viruses are encoding, is very common for viromics, and we have seen even in recent studies this can go up to 80% of reads in virome data. So how this viral dark matter can impact gut microbiota is largely well assumed through their bacterial communities, and this goes through the aspect of phage life cycle, whether it is lytic, in which case phage infects bacteria, it propagates, and then it releases pyrogenic viruses. In second option are the phages that are temporal phages or lysogenic phages. So these phages, they integrate, they detect bacteria and they integrate into the bacterial chromosome. They can replicate together with bacterial chromosome, and as such, they can also become very important decision-making switches in bacteria. They can encode different toxins or give bacteria novel capabilities. In certain instances, especially whether it's spontaneous and in case of stress, these phages can actually induce and produce functional phages, which can, again, infect new bacteria. So overall, role of bacterial shaping gut microbiota, we can say it's direct, where we have direct phage predation, which is not just common in gut, but very common in nature. And we have horizontal gene transfer from lysogenic or lytic phages. And it can be also indirect. We know that the phage disease can generate different metabolic landscape, and these changes in metabolic landscape can actually change the overall community, even bacteria that are not infected by the phage. So the implication of phages for the overall bacterial community can be quite high, and this has been shown to also be reflected in several studies in which phage communities have been shown to be differentially abundant or have differential community composition in different diseases, or in some cases even before onset of the disease. So what is important for us for phage actually in its abundance is how they actually behave on this mucosal layer. So some studies have reported that in this mucosal layer, phage outnumber bacteria in 20-1. And some other studies have also reported that there is augmented encounter rate with bacteria there, which is somewhat reasonable if the phages are in such a high number. So this means that phages could play also important role in addition to IgA, mucus, and antimicrobial peptides in order to keep bacteria and mucosal layer protected from these bacteria, commensal or pathogenic. Now, another aspect that we were interested here is this microbe-immune interaction, which are quite intense in this mucosal layer, and in which immunoglobulin A is the most dominant antibody in the gut or in human body plays a role. So we know that IgA plays a role in actually identifying pathogen or growing bacteria, but it also plays a role in positive interaction between bacteria. Now, in the absence of IgA, a condition that we know as IgA deficiency, pathogen can become closer to this epithelial layer, which can lead to overgrowth, to inflammation, gut dysbiosis, and it can also lead to bacterial translocation and more severe consequences. So these conditions are actually known as primary antibody deficiency. The one that I mentioned, in case we have a lack of IgA, is very frequent. It occurs 1 in 500 Caucasians. Another condition which actually happens when we have lack of IgA or reduction in IgA is common variable immunodeficiency. Now, this is all characterized by the lack or reduction in IgA combined with reduction or absence of IgA and IgM. So we wanted to look into these conditions because both IgA and IgA deficiency and common variable immunodeficiency have a broad range of symptoms, ranging from pulmonary disease, GI disorders, but some subjects can also develop more severe, such as autoimmunity, fatal liver diseases, or malignancy. So far it's not very clear why one group of individuals stays stable while other develops more severe consequences. I also have to underline that IgA deficiency in some cases has no or very mild symptoms in approximately 30% of the patients. So it is not really clear yet what completely balances the microbiota in such absence of IgA, whether there are other compensatory antibodies or such other mechanisms. So what we wanted to actually investigate whether any of these compensatory mechanisms is involving phages, or whether actually phages could be really important markers how we can differentiate between different groups, even in primary antibody deficiencies. So we benefited from direct access through the lab of Rashov to primary antibody deficiency cohorts. This cohort has been already characterized in 2018 by the lab, in which only bacteria were characterized. So we expanded on this by looking actually how viruses are changing in different populations. So we started to look at this in order to see actually if we can finally at some process actually identify also state of population that can protect from the disease, because our cohort of IgE deficiency subjects contained mildly symptomatic individuals, and they also have not received antibiotics in the past 3 months. So the way we approach viral characterization is somewhat different to general microbiome analysis. First, as I said, there is no common taxonomical marker for viruses, so we usually enrich the free viruses that are outside of the cells by eliminating cellular components, and then by eliminating DNA and RNA that is not encapsulated, that is free. We do this by DNase and RNase treatment. And finally, when we get these viral-like particles, we break these particles and we get the DNA. We can also do the viral study directly from, we do shotgun metagenomics. However, this approach does not give me only free viral particles, and it also has some bias in actually downstream analysis, but usually when we have opportunities, we prefer to do both combined together. In those steps, what we proceed is we do the assembly, and when we have the sequence assembly in large quantities, we have to identify which quantities are actually viral before we can proceed the final analysis. So this is somewhat important step that usually is not present in, for instance, bacterial or fungal analysis. So when we analyze this and when we finally got our sequences, we map the sequences to see what we have in our different cohorts. So we had control, as I said, IgE deficiency and CIMI, and we observed some more typical or dominance of caudiviralis in our data. And we look which specific viral family might be actually differentially abundant in our groups, because that was one of the questions that we wanted to answer. And indeed, we were quite interested that, for instance, autographiviridae, immunoviridae were groups that were differentially abundant in one or both primary antibody deficiency group. So in this case, in the first case, there was an increase in the family of viruses that are, well, they are largely diverse, as most viruses are. But for us, it was interesting because these viruses are assumed to have more lytic properties. And for instance, increase in immunoviridae in IgE deficiency was quite interesting, because we know this group of IgE deficiency was not so severely sick group of people, and this means that some of these phages could actually be important in balancing also the total microbiota, which requires, of course, us for further looking into this. Now, again, these immunoviridae are mostly lytic phages, and some of them have been studied for this property in also in phage therapies. Some other interesting observation we also see, like microbiote, so they've been decreased only in CIMI, and this also corresponds what we see, what has been seen by other authors, for example, in intestinal bowel disease, where this group of phages is also decreasing in abundance. Then we look at some more specific phages. For example, we say, okay, cross phages, we know that in some conditions they've been assumed to increase, well, actually decrease, and they're supposed to be more abundant in human gut, and maybe some specific cross phages that are linked to expansion. So what was interesting for us is that we actually see the expansion and not the decrease in cross phages. Overall, like the most abundant one, they seem to be expanding, starting with IgE deficiency and then CIMI, where we have somewhat more predominant cross phages in this group. Now, as I said, this was interesting for us because IBD, these phages are somewhat decreasing, And what we went, because we only, in this case, we generated the viromics data, but previously in the lab that the genomics data was generated and that was published. So we went back to that data and we refiltered it to look at actually what is the general dominance of the phages infecting Bacteroides or phages, because the Bacteroides are hosts for these type of phages. So we refiltered this and we actually indeed observed that in overall and metagenome there was increase suspected of reads that are mapping to phages infecting Bacteroides, which are mostly Pras-like phages. We also look at other phages, and we see that the phages infecting Enterobacteriaceae are increased in abundance, which was somewhat a logic because Enterobacteriaceae are flourishing both in IgA deficiency and especially in CIVID. Enterobacteriaceae are also high target of IgA, and its absent compensatory mechanisms such as IgM does not really supposed to not bind very well Enterobacteriaceae. So this has somewhat logic in the increased abundance. But we also observe some indication that these phages actually are not just most abundant, but they might be actually increasing in overall diversity. So this aspect, how actually interaction between phages in bacteria is shaped in the presence or absence of IgA was quite interesting for us, and especially also because Bacteroides are not highly targeted by IgA. It has been shown that Bacteroides interaction with IgA helps their colonization of some species. So we assume that this expansion of certain phages could be seen also through what we've seen before in previous studies, that this evolution and expansion can be shaped by selective pressure, and IgA is selective pressure. So we went back and we thought about it. If we have interaction or no interaction with IgA, this could actually maybe completely differentially impact this phage bacteria interaction. Now we could assume that bacteria can be bound by IgA, but previous studies have lacked to show this IgA binding of specific gut phages, and this has not been yet elucidated. So we went further back and we developed a method so we can actually study this, which consisted, of course, of taking the gut microbiota. We performed size selection to get free viral particles that are not linked to bacteria and other microbes. Then we stain these particles because we also are very small, so we can fluorescently stain them, that we can easily detect them on flags. And then we studied for binding or no binding of these phages by simply sorting it and sequencing it. And we were actually quite, so in this step, we can study phages naturally opsonized by IgA, such as in healthy human gut. But in case of IgA deficiency, for example, we won't be able to see this. So we can supplement with IgA externally here, or we can supplement with IgG in serum. So there are certain options that we can study. In our first attempt, we just looked at the natural protein in healthy gut, and we actually observed that phages virum binds IgA. And we also see that if we supplement with IgG, we can also see the IgG binding. Now, we have more IgG assuming because we have this purified IgG perhaps for multiple individuals. And finally, we perform sequencing. Now, as we have very little DNA from viruses, we were also quite happy to see that actually what we sequenced were viruses. And we observed what was also typically seen for IgA-bound bacteria, that not the same bacteria are bound. So we see this also for virus. It seems that IgA targets specific subset of viruses. And we were interested to see that also some of these viruses are Pras phages. And otherwise remains to be still characterized that, for example, these uncultured phages and so on. So to link this with IgA deficiency, we also performed similar assay. In this case, we look at the binding of IgG, which we supplemented to see whether these phages are similar phages we are detecting still in this cohort. And we indeed observed that also in IgG deficiency, we still see some Pras phages that are in positive fraction. And these phages are, we can see their reads or contigs all over their genome. And we also see other Bacteroides phages with really high abundance of reads, meaning that they are quite well present and well sequenced in our data, indicating the possibility of our methods to detect this virus binding IgA that has not been studied in the gut so far. So overall, by doing this method, we switched from this linear concept that we have a phage that targets bacteria, and then bacteria interacts with our immune system and antibodies, to this tripartite interaction, more complex interaction, in which phage can both interact directly with bacteria and with IgA, and this forms one tripartite interaction that, in this context of IgA presence or absence, could have important implication on the evolution of specific bacteria and phage. But in a larger context, this also could have important implication when we're studying different diseases, or when you're specifically trying to apply phages in phage therapy, whether specific IgA can actually be a limiting factor or binding, or whether phages could actually accumulate in different organs and systems. So as a final conclusion from the results, we could see that some specific phageon signatures and little phage predation in the absence of IgA in primary antibody deficiencies. Some of the consequences of human antibodies that bind to prevalent gut phages could be linked also to absence or presence of IgA. And this IgA preferential binding to phages, of course, warrants further perspective in studies into these tripartite interactions between IgA, phage, and bacteria, both in these disease conditions and phage therapy. So I would like to acknowledge finally Guy Goroshov and the team. So Simeon, who did the part of data analysis, Darine and Eva, who did the part of sorting with Christoph, and to access to cohort with Jean, Delphine, and Claire from St. Louis Hospital, and of course the funding. So with this, I would be very happy to answer your questions. Thank you very much for the attention.
Thank you, Lejla. Nathalie, je te passe la main.
Okay, thank you, Lilia, for this very nice presentation with quite new data. And we are not really used of managing a phage in this FHU, so it is quite a new topic and leading very new interesting aspect. I'm looking, but I don't see any question from the audience, so do not hesitate to ask the question. You can ask in French and we will try to translate, or maybe Lydia, you can directly answer in French, I can imagine. I will have a first question from my point of view. You know, I'm very interested in fecal transplantation and in bacteriotherapy. And your last slide say that it should be interesting to follow the place of the IgA with the bacteriotherapy. Could you develop?
Yes, so I think, like when you have a phage therapy, IgA is a limiting factor. So IgA binding to phage could be a limiting factor actually in phage therapy that has been shown for some phages. So there's one aspect, and the other aspect, like phage antibody could form a complexes. We don't know whether they go into organs, tissues, all this aspect of the safety of phage therapy. So this is another aspect that I said it would be interesting from this. And maybe the third aspect, which we have not answered, is this fact that this IgA deficiency subjects that are not very symptomatic, they might have very interesting viromes that could be applied in phage therapy, because they have been trained, they have been evolved to target pathogens. And this is something we know, like with IgG, that from IgA deficiency, it's more specialized in targeting organisms than from normal subjects. So maybe this group of asymptomatic IgA deficient subjects will actually be carrying very specialized virome for this phage therapy.
So maybe it could be interesting to select the patients in order not only on the safety, but on the basis of their global pathology.
Yeah.
Maybe I have also question, some actually very practical, maybe naive question. I guess the first one is the way we collect, this has been discussed a lot, is the way we collect stools in intervention. So is there some kind of specific recommendation we could envisage to be sure that we will capture some virus? Do you know? I guess there have been some tests on that already.
So would this be for purpose of propagating virus or just like the general?
No, just to study. I mean, as you are doing in your sample, is there? Because some people, for example, who are doing metagenomic data, mostly focusing on bacteria, can use also preservative solution to avoid degradation. So I guess the question is that is it the same to then study the virome, or is there some specificities we need to take care about?
I think preserving them at minus 80 is quite okay because they are quite stable. Then if you take them out, then it's good to have some calcium and magnesium salts, but it doesn't require anything. And as also they are not cell, they can be easily propagated, so it should be okay. We just need larger quantities than for bacteria because they are very small, but that's all.
And to complete the question, does the method that you use for the analysis normalized? Do we have a method that can be shared by all the scientific community, or do we still have methods which are lab dependent?
Yeah, so I think that we are always updating the methods because they are just developing very quickly, and usually each step we try to test, currently available methods for metagenomic assembly for viruses is this one, and we use the most recent one that gives us the optimal results. For identification of viral viruses from countries, we usually apply combination of methods, so we don't rely on one, we apply what is available. So the previous one that we have plus the new one. So for in our last study, we applied 5 of them just to identify the most from the viral group. So we usually just building on this, and yes, I mean I think most of the methods that are there are actually benchmarked and already published. So just how you combine them and how many you are using.
I guess there is two questions. So first is from Luisa de Sordi. So she mentioned you did a lovely talk, and maybe so here it's focusing on your IgA deficiency cohort. Did you do phage host prediction? If so, do you see phage change related to bacterial changes in relative abundance?
We have not done this resolution yet. We have to go back and do metagenomics as well for this. So we are still on that, and we have trying to implement the recent method for this prediction. So not yet.
I have, before the other questions which are coming, I have also a quick one is on annotation. Is there, as you know, I mean, for bacteria, microbiome, the catalog are rapidly, the genes catalog are rapidly evolving. So, some years ago we had like minimal catalog, now we have huge catalog, and we can annotate more and more. So, what is the community, the status of the community for phage? Does this progress? Can you annotate more and more also?
Yes, actually you can. So there are also more databases that are environmental databases, such as the one that I mentioned, that they catalog all the viruses that are from that are in metagenomes. So we can go from there. But then again, functional databases are the same for bacteria and for viruses. So we use the same databases to predict proteins and their function. So this does not change for, it increased together, I would say. So it's still a huge difference. We only have a really very huge difference in reference genomes in phage that are isolated than bacterial. So this has not been corrected, unfortunately, yet.
So maybe if we continue on that metabolic function, we have a question from Simon Platini, who say that do you think that metabolic function that are lost during disease can be restored using lysogenic phage targeting the specific bacteria? And is the gut viral necessary for the gut homeostasis?
So the first question, I'm not a huge expert in metabolomics, so I guess with one page you can't really do that, I would say, so that would be very difficult. And the second, of course, phages are part of gut microbiota. You cannot take phages out. If there is a bacteria, there is a phage. So it just, we often take a prism that bacteria are before phage, and we also take phages gene to be bacterial. So I think we are a bit biased towards, but definitely not possible.
Okay, and so let's move to a question about Clostridioides difficile from Nadine Kassir. And he said that IgA against Clostridioides difficile have been rarely studied, but may have a role in individual humoral response during infection. Do you have data on Clostridioides difficile phages and or IgA or IgG against Clostridioides difficile phages?
No, we have not been looking specifically into Clostridium, but we are still currently generating even more data, so we could have a look at it in the final analysis, I would say.
Maybe to follow up, so Marie Espati is asking about the difficulty for viral analysis, which could just reflect modification of the microbiota composition as a whole. And just a little, I may add that I remember some talks about the loss of microbiome diversity in the context of metabolic disease, and a colleague from Ireland mentioned that actually the loss of virome diversity was also a very strong signal in that case. So I think this is linked to the point. And so then the question of Marie-Anne Espati is, can you discriminate phage that would be specific of the IgA deficiency?
We have not gone to this specific resolution yet, because we are trying to integrate also large nanopore data, so we can characterize phages in really high resolution in IgA deficiency. And we are also intending to cultivate the phages that propagate in, for example, Enterobacteriaceae, to see if we reduce this population diversity, whether we can find some specific phages. There is some indication that some more phages, like for Enterobacteriaceae, do, from phages infecting Enterobacteriaceae, but I think we still require some validation for this analysis. There seems to be much more diversity, but the resolution still needs to be improved for the data, I would say. So it's very difficult to be so specific at the moment, I would say.
So we have a question now from Jeremy Welch from the Aster, and he said you showed that you can stain the phages, analyze them using flow cytometry. Have you ever tried to sort and subculture the phage in order to characterize them in vitro? I think that you already answered a little bit of that question.
Yes, we did. We did that. We cultured them. We add externally IgA, IgG, and specifically for Microbiote, and you can see that this is binding some antibodies. So it was possible. We had to account for some fluorescent background in the media, but it works equally well, as even better than what we have from fecal sample, because it's standardized, so background fluorescence is always the same, contrary to fecal sample where you have individual differences and you have to account for. So yeah, we did that.
Okay, and did you face some specific technological limitation?
Well, there are some technological limitations, like when we had to clean, for example, phages from, like, we wanted to like eliminate the external DNA before getting the phage, but then we realized if we are doing this, we also eliminating the IgA, so we had to compromise, and we also have to see that we are not interfered with the background fluorescence of each sample, and these kind of things that are very common when you work with environmental samples. So yes, but I think we have been on a good path to eliminate this and finally get this sequence data, which again, like for viruses, it's very difficult to go and directly sequence. So we managed to do this. But it was we managed also to overcome some obstacles along the way.
Okay, so maybe it would be interesting for him to discuss directly with you in order to answer all the different aspects of this question. So thank you once again, Lejla, for this nice presentation and a really new topic which was never discussed during this webinaire. Un grand merci, Lejla, pour ce talk, cette présentation qui était très claire et qui nous a permis d'aborder vraiment des questions tout à fait nouvelles qui vont peut-être ouvrir des nouvelles perspectives et des nouveaux axes de travail. Je vais terminer peut-être en parlant des 2 prochaines présentations dans des axes très différents, encore une fois, dont les problématiques sont larges. Donc le 5 février, ce sera Anne-Judith Radigora, qui est professeure à la faculté de pharmacie de l'Université de Paris, qui va nous parler de dysbiose et allergie. En partant de l'établissement du microbiote pour aller vers la modulation de l'écosystème intestinal. Et le 4 mars, le professeur Mouna Nachi qui parlera de la problématique de l'anorexie mentale et une fois encore du rôle du microbiote intestinal dans cette problématique.